Today's speakers are Chris Mell, Anna Henry, and Linda Hughes. Chris Mell is the Customer Support Manager and a software trainer at Certara. Anna Henry is the Executive Director of Certara University's Scientific Training and Education Department, and Linda Hughes is a Principal Software Engineer with Certara. Chris, Anna, and Linda, welcome to today's presentation. I'll now hand it over to Chris to begin the presentation.
Thanks, Suzanne, and welcome to everyone to today's webinar on RSABE for NTIDs. Today's agenda will include an introduction to the reference-scaled average bioequivalence method. We'll cover some of the regulatory guidances and methodology for narrow therapeutic index drugs, and introduce the Phoenix template project for RSABE for NTIDs, followed by a demo of the template. And we'll also inform you how to download the project template from Certara University. And at the end, we'll wrap up with a Q&A session. So a bit about the methodology. So BE refers to bioequivalence, and this is required by regulatory agencies for approval of generic drugs or differing formulations. A test drug or formulation must be proven to be bioequivalent to an approved reference drug or formulation for PK parameters such as AUC and Cmax. And typically these are log-transformed PK parameters. In average bioequivalence, or ABE, the 90% confidence interval for the geometric least squares means ratio of the test formulation over the reference for AUC and Cmax must fall between 80 and 125. The RSABE stands for reference scaled average bioequivalence, and this methodology refers to widening or narrowing of acceptance limits, where the extent to which the acceptance limits can be widened or narrowed depends on the intra-subject variability of the reference product. So an example of the widening of the limits refers to highly variable drugs, and narrowing applies to the narrow therapeutic index drugs. So why would we use a reference-scaled ABE for narrow therapeutic index drugs? Well, these drugs have a narrow range between therapeutic and toxic dose levels. Traditional average bioequivalence methods may be unacceptable for NTIDs because small differences in drug exposure, for example 20%, may lead to serious therapeutic failures or adverse drug reactions. The usual 90% confidence interval limits of 80 to 125 are not considered sufficient for NTIDs, and several regulatory agencies have narrowed the limits for bioequivalence, typically from 90 to 111%. Many regulatory agencies have issued guidances and guidelines specific to NTIDs to define the RSABE methodology, including the FDA, EMA, Health Canada, Japan PMDA, and others. The U.S. FDA Guidance for Warfarin Sodium, issued in 2012, proposed a new bioequivalence methodology for NTIDs, which includes RSABE to scale bioequivalence limits to the within-subject variability of the reference product and compares within-subject variabilities of test and reference products. Multiple other FDA guidances have also referred to this warfarin guidance, and these are listed here. The warfarin guidance states that a 4-period, fully replicated design must be used for NTIDs, which means that both the test and the reference drugs must be given more than once. An example design is TR-TR-RT-RT. The test formulation must then pass the following 3 criteria. The first criterion, RSABE, scaled to the reference variability. The following confidence bound, the upper bound, is computed using the chi-square distribution and must be less than or equal to 0, where the 95% upper confidence bound is the difference between the test and the reference squared on average, minus theta times the variance of the within subject for the reference. And this must be less than or equal to 0. And SWR, this is a term we'll refer to several times here, but that's the estimate of the within subject standard deviation of the reference product. An example of the first criterion for reference formulations where the coefficient of variation within subject for the reference is approximately 10%, the 90% CI must be within limits of 90 to 111.11%. And we see over here on the plot here where we have the 90% lower and upper confidence limits on the y-axis, and the reference variability expressed as a CV% for within subject for the reference formulation on the x-axis. So if we have a low within subject variability for the reference, we have a very tight confidence interval, and as this increases, the confidence bounds also increase. So sort of a classic example is if we have a 10% CV, the limits are between 111.11 at the upper end and 90% at the lower end. These continue to widen until we get a CV% up to 21.42%, after which we encounter the second criterion where the FDA recommends using traditional unscaled ABE within 90% CI limits of 80 to 125. So for larger reference variability, we have a cap. We no longer are widening the confidence interval, and we're simply using the traditional 80 to 125 limits. The third criterion is that the upper limit of the 90% confidence interval of the test to reference ratio of within subject standard deviations, sigma WT divided by sigma WR, must be less than 2.5. The confidence interval of this ratio is estimated as shown here, where SWT and SWR are the sample standard deviation estimates with V1 and V2 degrees of freedom, and F refers to the F distribution with an alpha value of 0.1. What about some of the other regulatory agencies? For EMA, Health Canada, and Japan PMDA guidelines, only an average BE analysis is required. Average BE is already provided by default in Phoenix Win Non-Lin, so a special template project is not required. However, these agencies do have tightened 90% confidence interval limits, and these must be applied to the ABE results. So for example, in the EMA, a case-by-case basis, but generally narrowed from 90 to 111, Health Canada, 90 to 112 for AUC but not Cmax, and PMDA, 90 to 111 for AUC and Cmax. This is the template project that we're showing today was presented as a poster at AAPS in 2017. And this poster is available for download when you access the template from Certara University. And it provides all of the details of the different criteria and all of the aims and objectives of using the template. So we've discussed the introduction and the regulatory guidances. Now I'd like to discuss a little bit more about the Phoenix template project for RSABE of NTIDs. And after that, we'll do a demo. So why are we using a template for this methodology? The RSABE can be formed in Phoenix using a reusable template project with workflows per the FDA guidance. The template project requires minimal input from the user and can be used with any input data set from a replicated 4-period crossover design. So the big advantage here is you just need to supply certain key information such as the test and reference compound names, whether or not the parameter is log transformed, and run through the template. So you don't need to be a programmer in order to use this. Phoenix Win Non-Lin provides a module to perform average bioequivalence, but this module is not currently designed to complete the other criteria needed for NTIDs, so additional tools must be used. The Phoenix template project, showing an executed example, is available for free download at Certara University. I'd like to discuss a few template project considerations. The template project was developed for Phoenix Win Non-Lin version 6.4 and is confirmed to work in later versions, but not earlier versions. It also works in the current version, 8.1. A published example dataset and R code from BBAC in Austria forum were used for testing the template project. The Phoenix results matched the results from R code. The template project was tested by today's presenters, but did not follow the formal testing and validation procedures for Certara software development. The template project was presented at an on-site training at the FDA Office of Generic Drugs, OGD, in 2017, and FDA has access to it. When using the template, inside the template project, full PDF instructions are provided in the instructions folder. You can simply click on the file from within your project and click View in External Viewer to open the PDF. A brief overview of using this template project. So the first step you'll need to do is download the template from Certara University and save the project with a different name. Import and map your data set containing PK parameters from a fully replicated crossover study for an NTID. Specify the reference compound name as well as whether or not the PK parameters are already log transformed. And lastly, execute the project in Phoenix to get results. Here's some sample output from running the template. The template produces two final output tables, one of which is the subjects excluded table. So if some of the subjects have incomplete data for one or more of the treatments or periods, the subject may be excluded for part or all of the analysis. So an example, if we look at the first subject here, the first subject had 2 reference periods but only 1 treatment period. So this subject was included in the average bioequivalence and included in the within reference standard deviation computation, but was excluded from SWT because there was only 1 treatment period available. And since the subject had an incomplete design, they were also excluded from the RSAVE critical bound. Another just brief example, subject 10 here had no data available for either period, so they were excluded basically from everything, AVE, SWR computations, and the critical bound. So a full listing of any subject exclusions is provided by the template. The second table that we get is the final RSAVE results based on the 3 criteria. So the criterion 1, 2, and 3, RSAVE for the critical bound. All of the information about that is listed here, including the point estimate, critical bound, and SWR used for the computation. The second criterion, average bioequivalence, shows the ratio as a percent of reference with the 90% CI lower and upper bounds. And lastly, the third criterion for the upper 90% confidence ratio of SWT divided by SWR. With the upper 90% confidence bound, this ratio must be less than or equal to 2.5. It shows the ratio and also SWT, which was used in the assessment. Okay. So at this point, I would like to provide a demonstration of the template. So I'm going to share out my Phoenix application. And I'll just pause briefly here to confirm that everyone can see my Phoenix application.
Yep, we can see it, Chris. It looks good.
Great. Thanks, Suzanne. So I have the template project loaded into Phoenix. So the first area we have here is the data area. It has a README, and it also has some of the input steps that you use. And when you import data, the data gets imported to this data area. I'll return to this a little bit later. I would like to point out that we also have the documents area where you can access the glossary of terms used throughout the template and also the information, the FDA guidance on warfarin for NTIDs, as well as instructions for how to use the template. So to open the PDF, simply click on the PDF item and then click View in External Viewer. So as an example here, if you click on View in External Viewer, you can access the draft guidance for warfarin sodium, the PDF instructions for how to use the template. So these are all step-by-step instructions. And lastly, we have the glossary PDF that includes all of the terms from the intermediate calculations that may or may not be familiar. So this is good for your reference. Okay, so I'll return to Phoenix. Okay, so the first step is to bring in some data, and I have some sample data imported into the template. I'll take a brief view of the input data set. So Phoenix likes information, all data of the given type to be in a column format. So we first have a subject column, and then the period. Again, this is a fully replicated crossover, so there's 4 periods per subject. The sequence, which will be TRTR or RTRT. The formulation name, which is capital T for test and capital R for reference. And I also have the AUC, both log transformed and untransformed. Okay. And so to run this through the template, I am going to expand the workflow area. Now, all of the items are in red because it hasn't been run, and so we need to actually run these to remove the red color. So the first thing to do is go to the user input. There's a data link item here called user step 3, map data for 1 PK parameter. So this pink spreadsheet here was from a previous run. In the top area under data, I should probably review the information in steps 1 and 2. So in step 1, one needs to enter the test and reference names. As we saw when I was going through the data set, the names were actually capital R and capital T, so I don't need to make any changes here. But if they're named reference, like spelled out, I would need to type in the full word reference here and the full word test here. Or if they were referred to as different names, it basically needs to match how they're spelled in the input data set. And there's a comment field here that kind of guides you as to what needs to be put in these fields. For step 2, this is the transform flag, and this is where you indicate whether or not the PK parameter is already log transformed. So Y equals yes and N equals no. In this example, I'll go ahead and type in an N just to show you how it works. So we will go ahead and use the example where it's not log transformed. And now I'm ready to go to my data link, and I'll go to the Setup tab, and I need to bring in some input data. So this is my sample data. Okay, in this data link object, many of the fields automatically mapped because in my input data set, the names match the anticipated column names. But if you have subj or patient, you can map that to subject. You also need to provide the period, sequence, and formulation columns. These have mapped automatically. And then lastly, the data that actually will be analyzed are in this data field over to the right. And I'm not going to use the log transformed AUC because I said it's not transformed in the setup. And instead, I'm going to use the AUCs. So at this point, I'll go ahead and run the data link. And it's a good idea to look at the output of the data link to make sure everything looks okay. So all of the columns are present. They're populated with data that looks like the data of the type in the column header. Now this is a template, so I've already supplied the minimal user input to get this to run. And from here on out, I'm going to do some data prep steps. So this is executing one of the sub-workflows. And essentially executing each of these sub-workflows in sequence to produce outputs. And you can actually see at the bottom of the Phoenix application, there's a little status window that shows you what it's doing, and it will actually say that it's complete when it's all done running. So it says execution complete. So that was the first sub-workflow. The next sub-workflow provides computations for the first criterion for RSABE. And I'm not making any changes here. I'm simply executing the workflow. Okay, it says execution complete. Ready to move on to the second criterion for average bioequivalence. We'll go ahead and run that. Okay, so that ran. And then the third criterion for the upper 90 CI limit ratio test. And again, so the items where you don't want to make changes are marked as do not modify in caps. So those are the template components. Run the next workflow, which joins all of the results from the 3 criteria into a single spreadsheet. And we'll do a bit more data prep for the tabulation. Okay. And the last 2 items are the output tables that I discussed in the slides. So the first is table of subject exclusions. When you get to the results, you want to make sure you're on this result table with the Internet Explorer icon. And we'll notice that this is an empty table. So the first data set that I just demonstrated here actually had a complete design for all of the subjects. So no subjects were excluded from this particular run. And then I'll move on to the final table and run it. And switch to the result view. So the result view, I'm going to give a little bit more room to see this table in more detail. So for the criteria, we see that it did pass the reference scaled AVE, where the critical bound was found to be minus 0.0098, which is less than or equal to 0. For unscaled AVE, it also passed here. The confidence bounds were 93.9 to 103.35. And lastly, it also passed the ratio test for the upper 90% CI, which is shown here. The ratio in this case was 0.448, which is less than 2.5. Okay, so that was a first example. And just to show you that you can also use this template with other data, let me go ahead and look at a second example. In the second example, we have the same input columns, and we have a log transformed AUC, but notice that there's some missing data for some of the subjects and some of the periods. So how does the template perform when we use an input data that has some missing observations? So in this case, since I only have the log transformed AUC available, I'm going to go into my step 2 and set the transform flag to Y, because the PK parameter is log transformed. My test and reference names are still R and T respectively, so I'm okay there. And at this point, I need to go into my data link object and switch the data column to the log transformed AUC. And I also need to unmap this and bring in the NTID for missing data. So this is now mapped to the missing samples data set, and the log transformed AUC is mapped to the data field. I'll go ahead and execute the data link, and we're now ready to run this through the templates. Okay, so the data prep step is complete, and we'll go ahead and run the RSABE workflow. Okay, RSABE is done. We'll move on to average bioequivalence. Okay, that's been run. Upper CI ratio test. And we'll join the results. So as you can see, this truly is a template. There's only a few inputs at the front end, and other than that, you can just simply run through and quickly get your results. We're now prepping the tabulation, and now I'll run the table of subject exclusions. Okay, so in this case, we can now see a list of any subjects that were excluded from the analysis and why they were excluded. So if I were to spot check and look at, for example, subject 4 here, they have the sequence TR-TR. They only have one reference, but they do have 2 test periods. So they're included in the average bioequivalence, but excluded from the SWR, which also excludes them from the RSABE critical bound. But since they had data for 2 periods, 2 treatment periods available, they were included in the SWT calculation. And then we have another example. Let's look at subject 18 that has sequence RT-RT. And this subject had no data available for the reference, but 2 for the test formulation. They were included in the average bioequivalence, but they were excluded from SWR, included in SWT, and excluded from the RSABE critical bound. And next, I'll go ahead and run the final table. Okay. So in the final table, we see the results for the reference scale. So the first criterion is that it passed RSABE. The critical bound was found to be negative 0.0105, which is less than or equal to 0. For average bioequivalence, the 90% CI was between 94.68 and 105.96, so that passes average bioequivalence. And then lastly, the upper 90% CI is less than or equal to 2.5, so the assessment is that it passes the upper ratio as well. Okay, so that is the template demonstration. I'll go ahead and stop sharing the applications and return to the slides. Okay. So with the demo complete, I would now like to talk about how to download the Phoenix template project from Soteria University. So first you want to go to www.soteriauniversity.com and go to the PK/PD free learning area. So this is, again, a free access, and it's under the on-demand courses. It's 106 free learning under reference scaled average bioequivalence. So once you click on the course icon, simply click subscribe to access the material. From there, you will be asked to supply your username and password if you already have a login to Soteria University. If not, if you're a new user, you just simply have to create a login with just some simple information, a user word, email, your first and last name, and a password. Once you get to the course content, there's a video player that has an instructional video showing how to use the template. And then just below that area, there's something called the file repository area that has the different free bioequivalence templates that we offer. And these are zip files. So you simply click the little blue link here, and you'll be downloading a zip file with everything you need to run the template. So this is a zip file, so you will need to extract the file, and it contains the project. And in the case of the NTIDs, it also contains the poster. There's also a sidebar that you can use to navigate for within this course. So you can look at the different videos and course content for other templates, such as the RSABE for highly variable drugs, along with the RSABE for neurotherapeutic drugs. Okay. And once you get the template project extracted, of course, you can access it just as I showed in the demo. So this is a snapshot of the project. And once you've actually created a login, after you log in a second time at Soteria University, you may need to scroll down to the My Courses area to get a filtered view instead of looking at all the upcoming courses. So this is basically a schedule of all the upcoming courses. Then if you scroll down to My Courses, you can access the ones that you're actually registered for. Okay. And then you would simply locate the 106 free learning to access these templates. I would like to comment that this is a growing area for us, so we will be developing more templates in the future for these bioequivalence analyses. The next one, we have a poster accepted for AUPS in 2018, and that's based on the FDA guidance for methylphenidate hydrochloride. using Phoenix Win Non-Lin. And this type of, so methylphenidate hydrochloride is a multiphasic modified release formulation for the treatment of ADHD in school children. And it's designed to achieve both a rapid onset of activity as well as sustained activity with a duration of 12 hours. So therefore, in addition to the typical PK parameters for bioequivalence, the guidance also recommends that partial areas under the curve metrics are calculated in addition to ensure that the generic formulation is therapeutically equivalent to the reference. So this these additional PAEC metrics are intended to better characterize the systemic exposure for early onset, sustaining the response in the middle of a once-daily dosing interval, and for maintenance of the response in the late stage. Okay? It also recommends to ensure the switchability between the brand formulation and generic products by using an individual bioequivalence method. So this is upcoming later in 2018. So that concludes the planned presentation. At this point, I would like to open up for the Q&A session.
Okay. Thanks so much, Chris. We invite our audience to submit your questions for Chris into the Q&A panel. So it looks like we've got our first question from our audience. Someone would like to know, can this workflow be used for partial replicate designs?
This particular template cannot be used for partial replicate designs, and the reason is that the guidance requires a fully replicated crossover design. In particular, the third criterion, where you're comparing the reference standard deviation of the test to the standard deviation of the reference. To get a standard deviation, you need the multiple observations for each formulation. So for this particular example, you cannot.
I want to know, can I use these templates if I have several PK parameters in a column?
Yeah. So if maybe you have Cmax, AUC, LAS, AUC infinity, they're all in a column. The answer is you can't right out of the box. What you'll need to do is do some data prep because the template's designed to only perform an analysis for one parameter at a time. But there are a number of data tools available in Phoenix that allow you to get it into a format where there's only one parameter per column.
Chris, someone wants to know whether these templates can be modified.
Yeah, so you can modify it. If you remember during the demo, several of the items in it were marked do not modify in caps, so you wouldn't want to change anything there. But you can actually add NCA objects or plots if you want to make some plots to the template. I remember when giving the course at FDA, their input data set wasn't in the same format that I showed in the demo. For example, they had an input data set where the times were actually the column headers, like hour 0 was a first header, hour 1 was a second header, hour 2 was a third header, and these columns contained the concentrations. So we did some pre-processing of the data set in a sub-workflow to stack all the times and concentrations into single columns, and then just added that as a workflow template to this template project. So it's certainly possible to modify, but it is important to not touch the items marked do not modify.
Answer earlier, so I think they don't want to set HBT.
Did we have another question?
Yes. So this is a question for my colleague, Linda Hughes.
Mm-hmm.
Can you demo partial replicate workflows, or is there a video demonstrating how to use them?
Hi, we previously gave a webinar on the highly variable drugs methodology, including RSABE. For those templates, you can use full replicates and partial replicates. So you can find that video on our website. We can get back to you later with the exact link that you would need to reference that video.
And Anna, did you want to comment on that as well?
You will have access to both recordings for this webinar as well as the one that we did last year, and you will get also step-by-step instructions on how to use them. So if you have a partial design for a highly variable drug, which is a little different than what we're talking today, you can use that design with the highly variable drug RSABE template. So just sign up for the course and you will get everything, as well as the upcoming one that we are developing. When it's ready after AAPS, we will put it as part of the same course. So you just need to register for free once, and you will get all these templates.
Thanks, Anna and Linda. Someone wants to know, why is the average bioequivalence output processed? Is it just an informative analysis?
Yeah, so that actually comes back. This is unique to the FDA guidance. So if we go back in the slides a little bit, when we talked about the different regulatory agencies that have different requirements, the FDA is kind of unique in this respect, in that we only apply the average bioequivalence when the reference variability becomes high enough. And that threshold is at 21.4% CV for the reference product. And rather than following the reference scaling bounds, which if we look in these extrapolated lines keep going out and keep widening, they actually cap it at the ABE limits. So even though we're using the scaling when the variability is low, and again, this is based on the warfarin guidance, they are capped at the traditional ABE limits when we get high enough variability.
Okay, looks to be the last question from our audience. Does the FDA use these templates?
Yeah, so the FDA has access to these templates, and they were presented at the training last year. We are not sure the extent to which they use them, but the FDA does have access to them.